Hello, physiologist. I'd like to talk with you about capillary exchange. Capillary exchange is how substances move into and out of your capillaries, and there are three main methods. You have diffusion, transcytosis, and bulk flow. Diffusion is the most important method, and it's just like diffusion that you have talked about anywhere else in the body. A substance will move from an area of higher concentration to an area of lower concentration. So if we are looking at your systemic circuit, for example, so all of your blood except for the lungs, you have a higher concentration of oxygen and nutrients such as glucose inside your blood and a lower concentration in the interstitial fluid which surrounds your cells, because your cells are constantly using that, right? So oxygen will move down its concentration gradient, move out of the capillary and into the interstitial fluid surrounding your cells. So now your cells have access to this new oxygen that's been delivered. Carbon dioxide is constantly being generated by your cells and other waste products. So you're going to have a higher concentration of carbon dioxide and wastes in the interstitial fluid surrounding your cells, and a lower concentration inside your capillaries, inside your blood. So carbon dioxide and waste will move the opposite direction. But again, they are still moving down their concentration gradients. Oxygen, carbon dioxide, everything else. Diffusion occurs along those specific substances' concentration gradients. So it's totally fine that oxygen and carbon dioxide are moving in opposite directions. They are diffusing down their own concentration gradients. And you may be thinking that glucose is kind of big for diffusion, and you are absolutely right, but there are channels that can help with facilitated diffusion. So still a passive process and still diffusion, but you still do have to follow those membrane permeability laws. So anything that's too big, anything that is polar, anything that is not lipid soluble is going to need a transport protein in order to use diffusion. Some capillaries are a little bit specialized, their anatomy is a little bit changed, so you may see a fenestrated capillary, you might see some intracellular clefts in particular areas of the body, and that would allow for a little bit more diffusion to take place, but you're not going to be able to move your plasma proteins. They are too big. And then when you look at the brain, you have the blood-brain barrier that is going to limit how much diffusion can take place and what substances are able to diffuse. It's much more selective in the brain because you have tight junctions that will limit that diffusion. Transcytosis is a combination of endocytosis and exocytosis. So you have a molecule, probably something that's just too big to move by itself. You have a vesicle that's created as you have endocytosis occur. And that substance, such as insulin, is kind of a big hormone, and it will move across the cell to the other side. As that vesicle merges with the plasma membrane, you have exocytosis occur. And so that's how you get movement of some larger molecules. Bulk flow is the last type of capillary exchange. And it is easy to confuse with diffusion because it also is based on a gradient. However, bulk flow is based on pressure gradients, not concentration gradients. And when you think bulk flow, I want you to think bulk, large, Sam's Club, Costco. You are moving a large amount of fluid plus a bunch of ions and molecules, particles, stuff that just kind of gets dragged along because you're moving so much fluid, they just can't help but tag along. Diffusion is still going to be the most important for solute exchange. So oxygen, carbon dioxide, glucose, waste, they get where they need to go because of diffusion. However, when you're talking about regulating volumes of fluid, that is going to be regulated due to bulk flow. And there's two processes with bulk flow. You have filtration and reabsorption. And these are just looking at the direction of flow. So filtration is when you have movement out of the capillary. Reabsorption is movement into a capillary. And these are due to different pressures, different forces. So filtration is going to be due to hydrostatic pressure. So let's review what hydrostatic pressure is. So hydrostatic pressure is often referred to as a pushing force. It is the physical force of a fluid being pushed against a boundary. So your plasma membrane would be that boundary. So you can see here in our U-shaped tube right here, you have a piston that is pushing down this fluid, and it's pushing against this boundary, or your plasma membrane. And as it pushes down, you can see the column moves toward the right side. And you have a higher column of fluid on the right side than over here on the left. So hydrostatic pressure is referred to as a pushing force in this way. And so if we look at how this looks in the capillary, as blood is flowing through on the arterial end of your capillary, you have lots of blood, proportionally speaking, coming in through the arterial end, and you have this fluid that's pushing against your capillary walls. And as this fluid is pushing against your capillary walls, some of it leaks out. So this is filtration. The process is called filtration, but the driving force behind it is called hydrostatic pressure. So the hydrostatic pressure is pushing this fluid out of the capillary and into the interstitial fluid surrounding your body cells. As the blood continues to move through the capillary, you come to the end of this capillary bed. Let's consider what you have left in the capillaries at this point. You still have erythrocytes, you still have leukocytes, you still have plasma proteins because they are too big to get out of the capillary. They're too big to be filtered. Remember that plasma proteins are very important for maintaining blood colloid osmotic pressure. Colloid refers to protein. And let's review what osmotic pressure is. So osmotic pressure is due to solutes, such as our plasma proteins, that cannot diffuse across a boundary. The boundary, again, is our cell membrane, our plasma membrane. So here you can see we have equal amounts of fluid on each side, but we have more solutes on one side than the other. Those solutes, and the solutes can't cross, so just like in the cell membrane, anything that's too big, like a plasma protein, or if it is not lipid soluble, if it's polar, you have to follow cell membrane permeability rules. So plasma proteins are too big, they cannot be moved across the capillary wall. So plasma proteins are here, and they are exerting a pull on that water. So water is attracted to those plasma proteins, and you can see they act as this pulling force. So osmotic pressure is often referred to as a pulling force for this reason. So on the venous end of the capillary, all these plasma proteins that are still inside your capillary are pulling almost all of that fluid that you filtered back into the capillary. And then that fluid, all of your cells, plasma proteins, will head back into this postcapillary venule and start heading back to the heart. So one more time. Hydrostatic pressure is the driving force behind filtration, which is what occurs at the arterial end. Fluid comes in, it's pushing against those capillary walls as it comes through the capillary bed. You have filtration occur. Once you get to the other side of the capillary bed, all those plasma proteins are going to draw most of that fluid back in. You've lost a decent amount of your blood volume at this point, right? Because you filtered it out. You filtered out a lot of that fluid. Plasma proteins will bring it back in on the venous side. And you'll actually reabsorb, so this process is reabsorption on the venous end, about 85%. The rest of it, the last 15% that is hanging out in your interstitial fluid, excess will be brought into a lymphatic capillary. So lymphatic capillaries are one-way, closed-ended capillaries that tend to follow along with your cardiovascular capillaries. So lymphatic capillaries take that excess interstitial fluid and they drain into larger and larger lymphatic vessels, and eventually those lymphatic vessels will dump all of their lymph, which is basically your excess interstitial fluid, back into your subclavian vein. So you'll get that fluid back eventually. It's just going to kind of take a scenic route. So one more time to sum up. Filtration on the arterial end is caused by hydrostatic pressure. Reabsorption on the venous end is caused by osmotic pressure, and that is thanks to your plasma protein, so colloid osmotic pressure to be specific. Here's another picture. showing a similar outcome. So here's your arterial end, blood comes in, and you can see there's some forces that are pushing out, some forces that are coming in. This force going out is referring to your hydrostatic pressure of your capillary, so that's your main driving force. You can see there is still some osmotic pressure that is bringing fluid back in, and that is totally normal. You have plasma proteins that will draw some of that back in, but your main driving force, you have more hydrostatic pressure, and that is going to promote filtration. So you have more filtration. As you move through that capillary and you've lost a lot of fluid to the interstitial fluid, you have some hydrostatic pressure left, although not much. The main driving force is osmotic pressure. All those plasma proteins will draw more of that fluid back into the capillary, and then as you move into the venule, you'll head back to the heart. If you have a disconnect, if you have a change in your capillary filtration or reabsorption, then that could cause edema. Edema is swelling. You have too much fluid in the interstitial fluid. And this could be due to a couple of different things. You can have too much capillary filtration, or you can have too little reabsorption. Both of those would end up with too much fluid in your interstitial fluid. Increased capillary filtration, so if you're filtering more than normal, think of things that will increase hydrostatic pressure. Increased blood pressure will increase hydrostatic pressure. More water retention, so increased blood volume, will increase hydrostatic pressure. So that's your driving force for capillary filtration. So anything that increases hydrostatic pressure will increase filtration. Also, if you have increased capillary permeability, so for example, in inflammation, increased capillary permeability makes your capillaries extra leaky. So it's easy for fluid to leave your capillaries more than normal. So you may notice if you get inflammation due to an insect bite, for example, you have edema in that area, and that is due to your leaky capillaries. You could also have decreased capillary reabsorption. So if filtration is just fine, but you're not taking enough fluid back in on the venous end, then that will have more interstitial fluid as a result also. And this could be due to a decreased concentration of your plasma proteins. So if you don't have enough protein in your diet, for example, if you are severely lacking in protein, you don't have the ingredients to make plasma proteins, that decreased plasma protein level is going to cause your blood colloid osmotic pressure to go down also. And if blood colloid osmotic pressure goes down, then capillary reabsorption goes down. So if you change the driving forces, if you adjust the hydrostatic pressure or if you adjust the osmotic pressure, you'll see changes in capillary filtration or reabsorption, and that could lead to edema.